Welcome back, everyone. So we just talked about the endoplasmic reticulum and the difference between the smooth and rough endoplasmic reticulum and the different functions and characteristics of those two parts of that organelle. And after the endoplasmic reticulum, you can see we have in this picture vesicles moving towards the Golgi apparatus. So this is how our membrane is flowing from the endoplasmic reticulum to the Golgi. So the Golgi network is functionally and physically linked to the endoplasmic reticulum by these vesicles. Within the endoplasmic reticulum, we'll take those glycoproteins and we'll take the lipids that have been made within the smooth and rough ER. And when they come to the Golgi, they're going to go through further processing. And then they're going to be sorted and packaged for transport to wherever they're going. Here they're just being exported from the cell. So the Golgi complex is important in membrane trafficking and protein trafficking. It makes sure that membranes go where they need to go, and it makes sure proteins go where they need to go. So the Golgi apparatus is the series of flattened membrane-bound cisternae. Kind of always reminds me of like just a stack of paper plates folded up, pushed up against each other. The cisternae are usually in a series of 3 to 8, called a Golgi stack. And some cells have one large stack. Others, especially if it's a secretory kind of cell, can have hundreds or thousands of stacks. Both the endoplasmic reticulum and the ER, both the ER and the Golgi, are going to be surrounded by all these vesicles that are carrying lipids and proteins from the ER to the Golgi complex and then to wherever its final destination is. So the Golgi lumen, or the inside of the Golgi, is part of the endomembrane system. Each Golgi stack has 2 different sides or 2 different faces. There is the cis-Golgi network here. This is the cis-Golgi network, which is facing towards the endoplasmic reticulum. Another side is the trans-Golgi network. Here, the trans-Golgi network is facing towards the cell membrane, facing away from the ER. Proteins and lipids are constantly leaving the endoplasmic reticulum in transport vesicles that are going to move towards the transface and then bud away from the trans-Golgi network. And between the trans-Golgi network and the cis-Golgi network are medial cisternae, and this is where most of that protein processing is going to happen. So each compartment is going to be biochemically and functionally distinct. Each compartment will have specific receptor proteins that are going to be unique to what that portion of the Golgi network is supposed to accomplish. There's 2 different models to try to explain how these lipids and proteins are moving from the Golgi. And one is the stationary cisternae model, which is what's in this picture. And in this model, we have the cisterna, and each of these stacks, each of the cisterna within this stack is a stable structure. So each of these stacks is going to stay here pretty permanently. But they create shuttle vesicles that move from one to the next compartment. So the shuttle vesicle will come from the ER to the cis-Golgi face, then just allow it to bud from one to the next to transport materials all the way to the membrane. In the cisternal maturation model, each of these compartments are transient. So the entire membrane is going to be moving and budding and bubbling down from the cis-Golgi to the medial cisternae all the way to the trans-Golgi networks. And then the enzymes that are needed for this, for this stack, as it moves, you'll just have these retrograde movement, allowing these vesicles to come backwards back to where they need to be. So the entire membrane is constantly moving, and anything that needed to be kept up in an earlier network just gets moved up through this retrograde movement. In both of these models, we are forming transport vesicles, and those transport vesicles are going to have sorted cargo that's supposed to move to a specific destination. And experimentally, the evidence suggests that these models are not mutually exclusive. There's a little bit happening from both of these in real life. So we've done experiments that involve tracking the movement of substances between these compartments, and there's also time-lapse fluorescence microscopy to be able to visualize how the movement is occurring through these different compartments. So we talked about two different kinds of movement. One is moving from the cis to the trans to the cell membrane, and one is moving backwards from the trans-Golgi network to the cis-Golgi network. So moving from the ER through the Golgi to the plasma membrane, it's called anterograde transport. So this is kind of that forwards moving from the ER through the Golgi to the membrane. So as these transport vesicles are being secreted from the ER and fusing with different components of the Golgi, that means that bits of the membrane from the ER are going to eventually travel and become part of the plasma membrane. But that movement of lipids from the ER to the plasma membrane has to kind of be balanced so that we keep a pretty consistent cell size, so we keep pretty consistent size of the membranes within our different organelles. So there's retrograde transport. Think of retro meaning like way back in time, like the 70s or retro. Retrograde transport also refers into going backwards. So this is vesicles flowing from the cytoplasm, from the cell membrane to the Golgi to the rough endoplasmic reticulum. So we have some vesicles moving anterograde and some vesicles moving retrograde. This lets your cell balance that flow of lipids to and away from the plasma membrane, and lets you make sure that you always have enough materials here in the ER to create new vesicles to carry materials forward again. The endoplasmic reticulum and the Golgi apparatus are both involved in protein processing. And within the endoplasmic reticulum and the Golgi apparatus, that protein processing is going to involve folding your proteins, quality control, making sure that everything's working the way it should, glycosylation, that is adding carbohydrate side chains onto your proteins, making them glycoproteins. There's two types of glycosylation, N-linked and O-linked. We've mentioned these before. N-linked means that it's getting added to a nitrogen, which is on the asparagine side chains. O-linked glycosylation means you are adding the sugar onto an oxygen atom. That means the oxygen atom could be on the side chain of something like serine, threonine, and sometimes even tyrosine. Within the ER, our proteins need to be folded into their final shapes. Sometimes they need to assemble with other polypeptides to form a quaternary structure there. And there's a protein called molecular chaperones. Here's some different chaperones that are involved in helping make sure that those proteins are properly folded. I think of like a chaperone at a dance, making sure that, the students are dancing correctly, not, smashed up against each other, behaving inappropriately. And within your endoplasmic reticulum, those chaperones in that lumen are going to make sure that your proteins are not smashed up against each other, but are, appropriately interacting in the way that they should be. There have to be disulfide bonds made in certain proteins to keep them safely within their correct three-dimensional shape. And there's an enzyme called protein disulfide isomerase. Protein disulfide isomerase, you can imagine, is involved in making and breaking disulfide bonds. Those disulfide bonds happen between the sulfurs on the side chains of cysteine amino acids. Disulfide bond synthesis begins before the polypeptide is even finished being formed. And then being able to form and break those disulfide bonds allows the protein to find the most stable arrangement that can be found. The way that we can tell if we have unfolded proteins, we have something called the unfolded protein response, or the UPR. This involves sensor molecules within the membrane of the ER that are able to sense when there are misfolded proteins. And when they sense there's misfolded proteins, they shut down protein production so that we're not going to keep making badly folded proteins. And we're going to turn on the production of proteins that are required for proper protein folding and degradation. So those proteins that are already made, those polypeptides that are already made, will have more of those folding proteins around to make sure that those polypeptides are folded correctly. And ERAD, or the ER-associated degradation, will pick up any misfolded proteins or unassembled proteins and kick them out to the cytosol, where proteasomes can break them down. Kind of like here, we have these proteins that are not being folded right, kicked out of the ER, the proteasome breaks apart that protein. Glycosylation also occurs both in the ER and in the Golgi apparatus. That initial glycosylation is happening here in the ER. Initial steps of glycosylation happen on the cytosolic surface of the ER membrane. Then later we can finish it within the ER lumen. And all of those carbohydrate side chains initially are going to be made of a common core oligosaccharide. That core oligosaccharide has 2 acetylglucosamine, 9 mannose, and 3 glucose. All carbohydrate side chains. That's that initial sugar that is added to the proteins. So in glycosylation, first you start adding glycosylation as a dolichol phosphate, with an oligosaccharide carrier is inserted into the ER membrane. Then the GLC-NAC and the mannose groups are added to the phosphate group. So here we're adding these side chains. And the growing core oligosaccharide is then translocated by a flipase. So here, flip. Now we have it over here. Now that it's inside the lumen, we can add more mannose and more glucose. So we're growing onto here. The core oligosaccharide is completed and transferred from the dolichol to the asparagine residue of the protein where it's going to be added. So from the dolichol, take it off, put it onto this protein. And this core oligosaccharide can be trimmed down and modified in whatever way that protein is going to need it. We can further process those N-glycosylated proteins in the Golgi complex as they move from the cis phase to the trans phase and turn them into really whatever kind of sugar carbohydrate we need added on the outside of that protein. Those terminal glycosylations, which are these glycosylations that happen here in the Golgi apparatus, are variable. So we have a big diversity of sugars. They'll be different in different species. Even different people will have different kinds of glycosylations or different cell types. Terminal glycosylation involves removing a few units of this core oligosaccharide, sometimes nothing more, sometimes adding other stuff back to it. Sometimes more complex oligosaccharides can be created by adding this GLC-NAC or other monosaccharides. Sometimes galactose units are added by galactosyl transferase, and there's hundreds of different glycosyl transferases that can add different sugar groups to it. All right, so we are going to continue our discussion of what happens within our endoplasmic reticulum and our Golgi network, how we're kind of making different proteins and targeting them to their correct areas in our next video. I'll see you later.